Thank you to the organizers to provide me with the opportunity to attend this, to co-learn and to get to know the real impact of impact. So congratulations on that. I'm really impressed. I'm Karel Lagarde. My background is neonatology and clinical pharmacology. I live in a very small country, Belgium, where you can actually drive through, respecting the driving restrictions, in 3 hours, which is somewhat different from this country. And I actually said during coffee break that I forgot to check where Vancouver was on the map. And actually my wife explained me before I'm leaving that this is really much further down than Toronto, or so that's what happens. But I've been asked to talk to you a little bit about, let's say, real-world data and PK data to inform the design. And so it has already been touched on that pregnancy and lactation are specific transient physiological settings who in themselves may affect pharmacokinetics as well as pharmacodynamics, like, for instance, autoimmune diseases or diabetes. You could imagine that these thresholds are not only just simply extrapolated from non-pregnant women. I have nothing specific to disclose except the fact that off-label use will likely be discussed. And for the sake of time, I tried to restrict a little bit my message to 3 bullet points. And one is we can be very pragmatic. And that's a little bit also how I understood the previous talk. We can include pregnant women in already existing trials. But we can actually also make trials that we can include a lot of different type of drugs exposures during pregnancy or during lactation and still have one single trial agreed on by the ethics committee or the IRB board. Second, we should perhaps be more accurate and complete. There is a zillion of data out that are not as useful as we would like them to be. Statement like, we were not able to observe the drug in the human milk. Thank you, but it's not really informative if you do not add the quality of your assay. So how low could you measure that? What you have done, actually, we are not able to measure any concentration below limit of quantification of observation. That matters, and I will explain you to a certain extent why. And then the most important point was believe in science. Believe in multidisciplinary science, because together we are stronger. And we would like to show you a little bit with PBPK modeling and also even more with physiological-based modeling. We can actually create an in silico pregnant woman, throw the drug in, and predict how the drug will most likely behave. And that's an important link with the previous speaker, because if you want to do a study design, if you want to do a PK/PD study for a given drug during pregnancy, it's terribly important to a priori select the most likely best scenario. If you have a trial with 200 pregnant women and at the end of the study you see, well, this trial didn't show any difference, but actually we were given the wrong dose, it would be a pity. And we have done that. I will illustrate that. So we have learned along the way and we should implement that. First message, be pragmatic. This is something that we just started about a year ago. We call it the umbrella study. And actually any mother who is breastfeeding can be included in this trial. It doesn't matter which drug she takes. We can simply assess, do we have any information on this drug already? So we don't need novelty if we already have 30 or 40 cases described. Second, we can discuss with the parents or the subject, what do you prefer to do? Do you agree that we collect blood samples in you? Do you agree that we collect samples in your infant? Do you agree that we collect mother milk? And if so, what is reasonable? Taking also the scientific background, like for instance, elimination clearance type of ideas. And do you agree that we collect safety data in you and or your baby? And we have something like a tick box. Because sometimes mothers agree that the blood sample is taken in their infant, or perhaps there is a clinical indication to collect so. And then you can adapt your clinical indication to your clinical needs. Quite common, they prefer not to, and that's okay. But in this way, you can be very pragmatic and That's the very reason that we also published this protocol, because we thought, okay, this is just Belgium, 3 hours drive and you're through. But in fact, we already recruited patients in London. You take the train in 2 hours, you're in London. A mother was taking clopidogrel, contacted us and say, well, I would like to contribute. And then we went there and collected all samples at her home. So my PhD student enjoyed 3 days of staying in London and came back with the samples. And actually, to put even more bizarre perspective, we have a patient now in Finland who has contacted a Texas group for advice on a given drug and say, well, we cannot help you. Well, they could advise, but if you would like to contribute to a trial, there's another group in Europe working on that. And from a US perspective, Belgium and Finland is close by. It's not really close by, and we have some burdens, but actually we recruited this patient and she's collecting every month. It's a very slow, very long elimination half-life compound. And she's collecting every month, and we'll go and get the samples somewhere in August. So in this way, you can actually be very flexible and ask parents also, what do you want? How far do you want to go? And what is a reasonable commitment for you? And like, for instance, the woman said, well, let's wait. I will do a first round. And if this is okay, I'm more than happy to give a second sample. And so this flexibility does provide much more comfort to both us as researchers, as well as to patients or families involved. So this works in our hands. It's a public domain, so you can simply take it and also do it in Canada or wherever in the world. The second point that I would like to stress is be accurate and be complete. And I simply would like to illustrate this based on a recent analysis, or relatively recent, on lactation and antidepressants, where a significant number of papers were excluded because it didn't provide sufficient information. And that's a pity, because I assume that the researchers who published this really wanted to contribute, and assume that the patients who contributed samples also wanted to be their contribution impactful. So how come? And I think even for, let's call it simple case reports, there's some guidance. The colleague Anderson has reported, or have provided, in my opinion, a useful suggestion. We try to stick to that. Report as much as possible data. What do you would like to see in a case report to make it as useful as possible, not only for yourself, but perhaps also for future pooling and making more sense of it. And my third and most important point is belief in multidisciplinary science. And as I was already alluded to, pregnant-related physiology changes, kidney function changes, body size and body composition changes, and also liver activities, enzymes change. This is not a dichotomous covariate. It's in general rather trimester mediated. So you're not pregnant or not pregnant, but it's a little bit more complex. But the good news is, and it's very similar as what we're doing now in neonatal, we can use this type of data. So even basic case reports of limited number of data would inform us. And just to illustrate it, I've never been pregnant myself, but I understood that a lot of women do not tolerate caffeine during pregnancy. Well, this can be explained because if you look well, on general, liver activities will increase during pregnancy, but there are some weirdos who do not, like CYP1A2, which is exactly the one who will metabolize caffeine. So you can exactly explain this. Your clearance goes down because you're pregnant. Hormone mediated. And so intestinal functions change, liver function change, kidney function change, not just GFR, but also transporters. But we have learned how they change. And again, you can actually make an in silico, so computer-driven system, where you can put all these covariates in. And you can actually also say, okay, this is a normal pregnant woman. Imagine that this is a woman with severe inflammation or obesity or other type of things. So you can add specific characteristics and further modulate your model to predict this more better. So there is a lot of strength in science to guide you in your clinical trials and actually to assist you in your study design. And there's a strength in it because we all know, for those who work in research, if you have a hypothesis, you can prove or reject your hypothesis. So if your hypothesis is, I have to give 1,000 milligrams instead of 800 milligrams to attain a similar exposure, you only need a few subjects to prove that you're on target. So that's the strength of this type of models. Again, we have learned in the meanwhile on the physiology and the changes throughout pregnancy, as is also illustrated on this slide, and we can use these type of things. Does it matter? The previous speaker also alluded to infectious mediated aspects, and this is another illustration going back in 2007 on a very rare antibiotic, amoxicillin. At that time, there was no guidance how to dose a pregnant woman with amoxicillin. And actually, this paper has been done because at that time, there were some concerns on anthrax. So sometimes infectious diseases or concerns, Max, we can also move a little bit the needle into the right direction for pregnant subjects. As you can see, this is the simulated exposure in the second, third trimester versus postpartum. I assume the majority of you know that you have to attain a given exposure to be an effective antibiotic. And actually, the normal doses are way too low for the second and third trimester of pregnancy. You can say, so what? Well, the funny thing is, for those who work in perinatology, they may be aware of the pivotal Lancet Oracle studies, who try to assess the impact of antibiotics for threatened labor and or without ruptured membranes, and whether antibiotics for 1 week were better or worse on the outcome of the pregnancy and the newborn. But post hoc, they simply used the wrong dose. It's unfortunate, but actually, post hoc, we should repeat these studies because at least what we know is that the exposure would have been way too low to have any therapeutic effect. And exactly the same is so for monoclonals, new hype in medicine, in human medicine and pharmacotherapy. We know how these monoclonals to a large extent behave. And in this way, different monoclonals, if you put them together, they always will learn you something on how does an average monoclonal will behave in a pregnant subject, and how can we translate it for even a drug that we never used until now in a pregnant subject. So that's actually the main point that I wanted to make. The placenta is a really interesting organ. It's not a coffee filter. It's much more complex. There used to be a time that people thought nothing comes across the placenta, but who would be so stupid because the fetus would never grow if nothing goes to the placenta, right? Then we thought it's just all passive diffusion, but there are a lot of things are happening over there: metabolism, active transport, active efflux, and we start to learn also on all these different type of expressions patterns throughout pregnancy. And in this way, you can predict, like for instance, for opioids, that if you have a genetic more active transporter or less active, that the exposure to the fetus will maybe likely different, and the likelihood of abstinence will be higher or lower. So we start to know more and more on how these different type of drugs behave. And this means that we can also predict to a large extent, based on the physical-chemical characteristics of this drug, how does this drug behave? How water-soluble is it? Is it metabolized? Is it by a transporter or whatsoever? You can predict with a reasonable likelihood how it will behave. And exactly the same, and this is something that I learned over the last years, is the beauty of the breast tissue. Because also over there, breast tissue is neither a coffee filter. You have, again, it's a little bit the way I try to teach to my students, say the breast is like a kidney. And then they look a little bit weird. But the point that I want to make is that at the breast, you have first passive diffusion type of patterns. And then you have a lot of modulations, just like you have in the renal tubules. Active secretion, active resorption, another type of stuff. So you can actually already predict to a large extent how much of a given drug, even if you don't have yet data, you can predict to a large extent what will be the most likely exposure. Exposure obviously doesn't equal exposure to the infant, because if the drug is not absorbed, it will not really make it into the baby. So a drug can be in the human milk, but may not be absorbed in the baby, and so therefore the consequences could be significantly different. I already alluded to this in silico type of models, and this is how a pregnant woman looks like. It's boxes. So you have, for instance, sorry. But you have a given blood flow to the lung and a given blood flow who comes out of the lung, and you have a given amount of lung, and you could have there a lot of enzymes who will metabolize your compounds. Same for kidney, brain, or whatsoever. You have a placenta, and you have all this type of stuff. And actually, you can average the weight and the size and the volumes. This sounds perhaps weird, but this is at present already commonly used for drug registration. If we discuss drug-drug interactions or drug-food interactions, a lot of companies will submit to FDA, EMA, or Health Canada this type of information. So this is not a new invention. This already exists as a concept in current drug approval and drug development. So to bring it to a next perspective, what if you can combine in vitro data, cell models, with in vivo models, animal models, with human findings? In this way, you can really learn much faster and have the best selection of a clinical trial. This is what we have done. Unfortunately, the study never took up because EMA decided that sildenafil can no longer be given to pregnant women, irrespective of the indication, which is, don't tell it anywhere else, but it's stupid. So we try to convince them that indication also matters in life, but you can actually predict how sildenafil will behave, and this way can predict how sildenafil will make it to the fetus. The background is we want to develop a new treatment for diaphragmatic hernia. And obviously, if you give it in healthy fetuses, as well as in animal models, their lungs are poorer off. In hernia, they are better off. So that's the reason that we want to convince still EMA that they have to reconsider their position. And exactly for pregnancy, you can also develop such models for lactation. You can have in silico, you can have in vitro models, cell models for transporters. You can have animal models. What we commonly use is the pig model, but there are also other animal models. And you can actually make sense from 1 or 2 case reports of clinical observations to bring all information together and estimate most likely scenario. At present, we are in negotiation. We will not make it. Negotiation with EMA to say, well, if you would use this model, you can actually predict once a drug comes on the market, this is how it will likely behave. So you can actually, with a reasonable scientific confidence, say the likelihood that this drug will be relevantly exposed in human milk is very low anyhow, which would be very informative for clinicians and families who have to make decisions. You can also use this for recreational type of things, like cannabis. You have a mother compound, you have a baby compound, you have a milk compound, and then compartment, and you can actually predict how it will behave, how it will appear in breast milk, and how it will appear also in the baby. So along these lines, the real world data, in my opinion, matters because it's really data integration, lactation-related infant physiology and postpartum maternal physiology who will assist you to make this type of lactation models more relevant. We have to be aware that the baby who's having breastfeeding is not the same average baby as a baby who receives formula. And so one of the things that we are currently working on is develop a model that is actually a breastfed baby. They grow different, but actually they also, that's quite intriguing, they metabolize drugs differently, as illustrated for, again, caffeine or dexamethasone. So no worries, I still have 30 seconds, and this is my last slide, I think. I hope to convince you that be pragmatic, develop clinical trials that can be applicable to different type of drugs instead of submitting a zillion type of documents to your ERB or ethics committee, whatever it's called. Be accurate and be complete because other people would like to use your data, and the more information they have, the more robust they can be used. And believe in multidisciplinary science. It may be weird for you, but science is there to support you and actually to develop your clinical trials as good as possible and have the highest likelihood to make it into a success. Thank you for your attention.